Hello, my dear audience, and welcome to another episode of letstalkmicro. As always, I am so happy you're here with me. And if this is your first time listening to letstalkmicro, welcome. This is a podcast where we try to explain microbiology terms as simple as we can. And there are some episodes where I go over organisms, and then there are some episodes where I have guests. This is one of the episodes where I go over organisms. And the last time I did this, I went over Eikenella corrodens, which is a fastidious gram-negative rod. And keeping it in that line, I thought I will go over another example of a fastidious gram-negative rod. So I'm going to be talking about Haemophilus. So let's go ahead and dive into it. And one of the things that can be challenging when it comes to Haemophilus is the gram stain. So if you're a student, I'm sure that you struggle with it. I know that my students sure do. And if you're working in the lab at some point in time, I'm sure you have had a blood culture. Let's say you find yourself alone, you make a slide, you look at it, and then the issue is, what is it? Is it a gram-negative rod? Is it a gram-negative cocci? I can't tell. You have difficulty, and this is something that I see with my students when we are doing gram stains and I give them Haemophilus, especially Haemophilus influenza. And they are looking at it, and then they either write for the test, they write gram-negative cocci, because it is hard, they are very tiny, cocobacillary, so you can have a hard time telling if it's a cocci or a rod. So this is something to keep in mind. If you are doing a gram stain and you see that it's gram-negative, and you have a hard time telling what it is, in all likelihood it is Haemophilus influenza. It can also be Pasteurella, but probably it's Haemophilus influenza. And I will talk more about this during the episode. But let's talk about some other things. Well, Haemophilus species, they are part of the Pasteurellacea family. They are pleomorphic, non-motile, facultative, anaerobic, gram-negative rods. And this is something that I just mentioned, so because of them being pleomorphic, that's why you can have that difficulty telling if they are cocci or rods. And most species of Haemophilus are part of the human respiratory flora. Haemophilus influenza can colonize the respiratory flora of healthy individuals. And according to the Manual of Clinical Microbiology from the American Society for Microbiology, there are 8 species that affect humans. These are Haemophilus influenza, Haemophilus aegyptus, Haemophilus ducreyi, Haemophilus pittmaniae, Haemophilus parainfluenza, Haemophilus hemolyticus, Haemophilus parahemolyticus, and Haemophilus paraphroghemolyticus. And during the episode, I will be touching on most of these, especially the classic ones that you will need to know when you are taking, let's say, a board exam like the American Society for Clinical Pathology or ASCP. And if you're a student, I'm sure that you probably suspect which ones I'm going to be talking about. But okay, let's continue. So there were some species that used to belong to the genus Haemophilus, but now are part of the genus Agaricobacter. And those are Agaricobacter ophiophyllus, Agaricobacter paraphroghemolyticus, and Agaricobacter segnis. And you know how that is, right? This is something that might drive us crazy in microbiology, but you sometimes have all these name changes, all these nomenclature changes, and sometimes as more testing is done, and especially with all this molecular testing, and then you realize that some organisms, they are differing enough to be reclassified, or maybe they're similar to another ones. So we end up with all these changes. And this is something that we have to keep up with, especially if we bring more technology to the lab and we bring things like multi-TOF-MS and gene sequencing. We're going to be seeing all these names. Okay, so we know it is challenging, but let's get back to Haemophilus. And I'll be talking more about media, but those of you that work on the bench and you work with respiratory samples, you know that you spend time looking at that chocolate plate, looking for those tan colonies for Haemophilus, right? That is definitely part of the workup, looking for that chocolate plate, right? Looking at it. And so, Haemophilus, they are fastidious gram-negative rods, and as you know, fastidious organisms have special growth requirements. And an example that I mentioned of this is Eikenella, which is a fastidious gram-negative rod, And in the episode, I mentioned that it does not grow on McConkey. Well, as the definition says, Haemophilus is not a stranger to growth requirements. And as I am saying this, if you are thinking of the X and V factors, then you are correct. And due to the requirements of those X and V factors, that is why Haemophilus does not grow on blood agar. And I will be talking more about that. When I talk about blood agar, I'm referring to sheep blood agar, but they do grow on chocolate agar. So let's go ahead and take a minute and talk about the X and V factors. The X factor, known as protoporphyrin 9, is an intermediate in the hemine pathway. And then the V factor is NAD, which, bear with me as I try to pronounce this, it is nicotinamide adenine dinucleotide. So how do you think I did pronouncing them? So let's continue. Both of these, the X and V factors, they are in the red blood cells. And think about that in chocolate agar. Why is Haemophilus able to grow on chocolate agar? And I will talk more about that. The requirements for X and V factors vary per species. For example, Haemophilus hemolyticus, Haemophilus influenza, and Haemophilus aegyptus require both X and V factors. Haemophilus ducreyi requires the X factor, and other species require the V factor. And I will talk more about this when I start talking about agar, about the X and V factors. Okay, so let's go ahead and talk about transmission and pathogenesis. For Haemophilus influenza, the production of capsules and factors are associated with virulence, and there are different serotypes. However, many infections caused by Haemophilus influenza are from non-typable strains. And let's talk a little bit about this. I'm not going to spend too much time on this. But the typable ones are based on capsular characteristics. The capsule is made of a sugar alcohol phosphate, and the differences in this capsule separates the capsular strains into different groups. And they are A, B, C, D, E, or non-typable. And this will make more sense when we start talking about the vaccine, right? For like type B. Or maybe if you do QC in the lab, maybe you have a strain that says Haemophilus influenza type B, right? So it will make more sense. Now maybe it makes more sense. Transmission occurs from person to person via respiratory droplets. Also, it happens when those strains that are colonizers gain access to sterile sites. Infection with Haemophilus influenza includes meningitis, otitis media, sinusitis, pneumonia, and bacteremia, among others. And students, keep this in mind. By now, I have gone over some organisms that are seen in meningitis, and this might be a question for a test or for your ASCP or your board exam. But by now, I have gone over Streptococcus pneumoniae, Streptococcus agalactiae. I also did an episode on listeria, so those organisms are seen in meningitis, so keep that in mind. And now we are adding Haemophilus influenza. So what about Haemophilus parainfluenza? So according to the Manual of Clinical Microbiology, Haemophilus parainfluenza is the predominant species that colonizes the upper respiratory tract. However, it does not routinely colonize the nasal cavity. As far as infections, it can be seen in otitis media, bronchitis, and sinusitis, but keep in mind that this is not frequent, right? So these cases are not frequently seen, even though they are possible. And remember HASIC? Well, what is the H in HASIC? And for the benefit of everyone listening, HASIC is, it's a group of organisms. Well, HASIC is the acronym, but it's a group of organisms that are seen in bacterial endocarditis. And please make the distinction that endocarditis, it is not exclusive to these organisms, but you tend to see the organisms in these cases. And Haemophilus parainfluenza is the H on HASIC. And once again, keep in mind that the cases are infrequent. Another significant species of Haemophilus is Haemophilus ducreyi, and this one is not part of the human flora. It is transmitted person to person via sexual contact. And this organism causes a sexually transmitted disease called the chancroid. and it is described as a painful genital ulcer. Another species is Haemophilus aegyptus. This one causes conjunctivitis. And then there are other Haemophilus species, as I mentioned, but when they cause infections, they are similar to those of Haemophilus influenza. So just to recap, with the exception of Haemophilus ducreyi, species of Haemophilus are part of the upper respiratory microbiota. And Haemophilus influenza is transmitted via person-to-person contact or via an endogenous strain gaining access to a sterile site. And other species can cause disease when endogenous strains gain access to sterile sites. Well, okay, so let's go ahead and talk about lab diagnosis. And here I'll start talking about the gram stain and media. So, gram stain first. As I mentioned, they are pleomorphic gram-negative rods, and Haemophilus influenza is more cocobacillary, or short rods. And that's why you have that difficulty, because they can be very tiny, so you kind of have to scan around that slide and look for a rod shape form. And this is something good for students or techs to remember when you're looking for those type of stains, right? Typically gram-negative cocci, like for example, your Neisseria gonorrhea, your Neisseria meningitis, your Moraxella catarrhalis, those are cocci shaped, so you don't see gram-negative rods. So what I mean is, typically, if the organism is a gram-negative cocci, all you're gonna see is cocci on your stain. So with the case of Haemophilus influenza or some species of Pasteurella, you might see something cocoid, very tiny, but if you scan around, you can see gram-negative rods. And that's an indication that you have a gram-negative rod. So keep that in mind. Haemophilus hemolyticus appear as short cocobacilli or short rods. Haemophilus parainfluenza can have pleomorphic or long rods. And for those of you that are studying for a certification, you might hear the term school of fish, and this is associated with Haemophilus ducreyi. You can also hear the term railroad tracks. However, according to Bailey and Scott's diagnostic microbiology, this morphology, the railroad tracks or school of fish, is rarely seen in clinical specimens. So from all this conversation about gram-negative, about the gram stain of Haemophilus, there are two things, and I said them, but I'm going to reemphasize them. And I want you to take home from this conversation. And one is, if you are reading a gram stain and find yourself having difficulty what it is when it comes to like a gram-negative organism, it is probably Haemophilus or Pasteurella. And the second thing, which builds, it comes from the first, is that if you find yourself with this difficulty and you see this tiny cocobacillary organisms on your slide, scan around and see if you see some gram-negative rods. And that's an indication that you have a gram-negative rod and not a gram-negative cocci. Because with your Neisseria gonorrhea or your Moraxella, all you're going to see is gram-negative cocci. You don't see any rod shapes. So as someone that's, that does what you do, the listener, that you work in the lab, I hope that this helps, right? So let's go ahead and talk about media. Well, as I mentioned, species of Haemophilus have varying requirements for X and V factors, with Haemophilus influenza requiring both X and V factors. Blood agar, and when you think of blood agar, you think of sheep blood agar, it has the X factor. However, the X and V factors, both of them, they are in the red blood cells. So most species of Haemophilus are not able to grow on blood agar. They are able to grow on chocolate agar, and this is because the blood in the chocolate agar is heated, which lyses the red blood cells, therefore releasing the X and V factors. Now, this brings the question, can Haemophilus grow on blood agar? And if you are thinking of the satellite phenomenon, you are correct. So, what is the satellite phenomenon? Well, if an organism can lyse the cells on blood agar, then both factors are released. So an example of this is Staphylococcus aureus. And the Staphylococcus aureus can actually, it produces NAD, factor V, as well, as a byproduct. So a hemolysin producing strain of Staph aureus in blood agar, if you have that there, then Haemophilus is able to grow around it because Staph aureus is producing NAD and is lysing the red blood cells in the agar, so both X and V factors are released. And this is seen, this satelliting is, you see pinpoint colonies gathering around or satelliting around the colonies of Staph aureus. And remember that it has to be a strain of Staph aureus that lyses the red cells in the agar. So keep this in mind when you are reading cultures and you're looking at your blood plate and you see Staph aureus and you see tiny colonies around the Staph aureus colonies. Make sure you check that chocolate and look for those tan colonies, which are representative of Haemophilus. And this is a very simple test to do, even though, and it is not recommended to do it like all the time, but it is very simple to do. And all you do is you inoculate, a blood plate with the suspect colonies, and then streak a line with that, that hemolysin producing Staph aureus and incubate your plate. And then the next day you should see the line with the Staph aureus growing and those pinpoint colonies growing around the Staph aureus because now they have all the things they need, right? Those X and V factors. And remember that CO2 stimulates the growth of Haemophilus, and you're thinking chocolate plate and you're thinking CO2 incubator. And that is why maybe if you're wondering when you go into a lab, you have CO2 incubators, you have non-CO2. And typically on your non-CO2, you have things like urine cultures. And then on your CO2, you have all these cultures that have chocolate, right? Like wound cultures, body fluids, respiratory cultures. So if you're trying to recover Haemophilus, the plates need to be incubated in CO2 conditions. And the colonies grow from 24 to 48 hours with Haemophilus ducreyi and Egyptus, taking longer, about 5 days, they might take longer. And something I mentioned is that Haemophilus, they are facultative anaerobes. So they can grow anaerobically. And something that is very interesting, I don't know if you have tried to ever plate Haemophilus influenza on a blood agar and then incubate it anaerobically. If you have, you notice that it grows unassisted, it doesn't need Staph aureus or anything. And I haven't done much research about this, but maybe, and don't quote me on this, I think that it might be with the pathway that they take to grow, like the electron acceptors and things like that. So that might be a reason, but it grows alone. And I find that really cool. But, that brings the subject of error tolerance. And when you talk about anaerobes, right, if you want to rule out an anaerobe, like if you want to find out if the organism is an obligate anaerobe versus a facultative anaerobe, you do something called an error tolerance test. And in this test, what you do is you take two plates, one is a chocolate, one is a blood agar plate, and you inoculate them with the organism, you incubate the chocolate aerobically, and you incubate the blood anaerobically. And of course, an obligate anaerobe is not going to grow on your chocolate, it's only going to grow anaerobically. But maybe now it makes sense of why to use a chocolate instead of a blood plate for your aerobic incubation. And that is because you don't want to miss the growth of a fastidious gram-negative rod, such as Haemophilus influenza. So by using that chocolate plate, you're ruling in fastidious gram-negative rods. And that way, if something is not growing on chocolate, it's a better indication that you have an obligate anaerobe. Whereas if you're using a blood plate for your aerobic incubation and nothing is growing, it brings the question, is nothing growing because you have an obligate anaerobe, or is nothing growing because the organism is not able to grow on blood agar? So, as I like to say, keep that in mind. And before I move to other media, I just want to let you know that Haemophilus can grow on liquid media, such as thioglycolate broth or brain heart infusion. However, it does so at a slower pace. Okay, so some recommendations for agar when it comes to Haemophilus include chocolate agar supplemented with 1% isovitalic X or Vitox. And this has vitamins, amino acids, and glucose, which provides necessary components to support the growth of Haemophilus ducreyi and Aegyptus. There is also Leventhal's medium, and this one I haven't worked with, but according to the Manual of Clinical Microbiology, this medium is transparent, and colonies can show iridescence, which is associated with encapsulated strains. So if you want to do more research on your own on Leventhal's medium, please go ahead and do so. But I'm going to keep talking about media. Okay, so one of the issues that we have when it comes to respiratory samples in the lab, is all the microbiota. If you work in the lab, if you work with respiratory cultures, you know that sometimes, those plates are loaded with organisms. And this makes it difficult to recover Haemophilus, right? Especially Haemophilus influenza. So there are some recommendations regarding agar that can help with this, and I'm just going to go over some examples. One of them is Haemophilus isolation agar with bacitracin and horse blood. And there's one that I found from Remel, which has no affiliation to the podcast. It has beef heart infusion and casein to supply nutrients. There is also horse blood in the medium, which provides the X and V factors, and it also serves as a differential medium between hemolytic and non-hemolytic strains. And as I mentioned in the lab, maybe if you work with sheep blood agar, you don't see these hemolytic strains, but this medium has the capability of that. Then the bacitracin inhibits the growth of other organisms that are part of the respiratory flora. And this is very easy to use. You inoculate your plate, then incubate it at 35 to 37 degrees Celsius in a CO2 incubator from 24 to 48 hours, and then you examine for growth. Then there's also a chocolate agar from Hardy Diagnostics, no relation to the podcast, no affiliation, with bacitracin. And this agar has something called coenzyme with a K, and this provides V factor and other nutrients. Then the heated blood on the agar provides the X factor. So it is a chocolate agar, so the blood has been heated. And just like I mentioned, just like the other agar that I mentioned, the bacitracin serves to inhibit the growth of respiratory flora. The plating and incubating conditions are the same. So you see with these two examples of agar, you're providing both the X and V factors, and you're also trying to limit the growth of respiratory flora. So that is the purpose, is to help you isolate Haemophilus, which, as I mentioned, it can be challenging sometimes. So to wrap it up, there is one more. And this one is the Haemophilus quad plate. And this plate is divided into 4 quadrants. Quadrant 1 has X factor only. Quadrant 2 has V factor only. Quadrant 3 has both X and V factors. And then quadrant 4 has horse blood, and it is supplemented with NAD. So it has both the X and V factors. And the medium has beef heart infusion to provide nutrients for fastidious bacteria. Okay, so now that we have talked about media, right, I have been mentioning things like tan colonies, and I have talked about the chocolate agar. So the next topic is colony morphology, which is what I'll be talking about in the next episode. I'll start with morphology and then move on to the ALA test, antimicrobials, and some other things. So stay tuned for that. And that concludes this episode, my dear audience. And as always, I hope you enjoy learning about this. But if you want to follow the podcast, it is available on all podcast platforms. So please subscribe and download episodes. And if the app allows you to do so, please go ahead and leave a review. And as far as social media, you can find Let's Talk Micro on Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and there's an email address which is letstalkmicro@outlook.com. So if you have any feedback, any possible topics, you can reach out with those via social media or via email. And if you want to support the podcast in any way, Let's Talk Micro does not have any sponsors. So if you want to support the podcast, you can do so via a Venmo link or via Buy Me a Coffee, KOFI, both links available on the show notes. And if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And these are some great resources by Dr. Timothy Gauthier, a very passionate pharmacist. And of course, Let's Talk Micro is supported by the American Society for Microbiology, so to learn more about microbes, visit asm.org. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about Haemophilus, and I'll continue this series. As always, I enjoy sharing this information with you. And hey, are you going to Microbe? It's approaching in Los Angeles this year in June. So if you're planning to attend and you see me, stop by, say hi. Thank you so much for the support. Stay tuned. Great things coming your way. So please, as always, continue bringing that passion to what you do. It is so important. You do such great work. So I am so grateful for the support. So as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.